Hi everybody, this is Sarah Red, and today we're going to be talking about enzymes and how they work. So first of all, what are enzymes? Enzymes are proteins, so just like other proteins, they're made out of strings of amino acids, and the DNA codes for the order of amino acids that will determine the shape of the protein. The special thing about enzymes compared to other proteins is that they catalyze chemical reactions. Catalyze means speed up, and a chemical reaction could be anything that rearranges bonds to build new molecules or break new molecules down or change their shape. Often the enzyme speeding up the chemical reaction can be a really dramatic difference. For example, there's an enzyme called catalase in all of your cells that breaks down hydrogen peroxide and forms water and oxygen bubbles. So if you've ever poured hydrogen peroxide on a cut and seen the bubbles, that is the result of a chemical reaction. Without the enzyme catalase in your cells, it would take about 1,000 years to break down the same amount of hydrogen peroxide as you get in less than a second with that catalase enzyme in your cells. Very dramatic. Most enzymes are going to have names that end in the letters ASE. So some examples would be lactase, which is the enzyme that breaks down the sugar lactose. Catalase, which is the enzyme we just mentioned that breaks down hydrogen peroxide. Even longer names like DNA polymerase is an enzyme that builds DNA into a larger molecule called a polymer. Each enzyme is going to have a unique shape, and it's going to catalyze one particular kind of chemical reaction. You have around 1,300 enzymes in your own body as a human, and there are many other kinds of enzymes in other species as well, each with its own unique shape. So we mentioned that enzymes catalyze chemical reactions. A chemical reaction is just a process that makes and or breaks chemical bonds. In a chemical reaction, the starting molecules are called reactants or substrates. These molecules will turn into new molecules that we call the products. When we write the name of a chemical reaction, we use an arrow to indicate the change that's occurring. Before the arrow, you will have the name of the substrate or reactant, and after the arrow, you'll have the name of the product. Next to the arrow, there will often be the name of an enzyme that catalyzes that particular reaction. So here are a couple of examples. The substrate sucrose is turned into the products glucose and fructose. The enzyme sucrase helps to speed up that reaction so that it will happen quickly. This is an example of a digestion reaction. Here's another example. The process of combining water with carbon dioxide happens in your blood. The enzyme carbonic anhydrase is going to speed up this reaction, and the water and CO2 will be turned into the product H2CO3, which is also known as carbonic acid. If you've worked in healthcare before, you might know that when people have labored breathing, their breathing is too slow, that carbon dioxide is not being exhaled fast enough, and so that carbon dioxide builds up at higher levels in their blood. And as that carbon dioxide combines with water in the blood, it turns into this acid, carbonic acid. And so one of the possible results of labored breathing is that the person can go into acidosis, which, if it's not dealt with, can be pretty severe. So let's talk about how enzymes work. So this large purple blob in the picture is representing an enzyme. On the previous page, I showed two depictions of the same enzyme. So these ribbon diagrams here are a more detailed and accurate depiction of what an enzyme is actually like. It's like a string of amino acids connected together. and that string folds back on itself, and parts of the amino acids that have positive or negative charges will bend to be near each other. But often, to simplify things, we will draw a two-dimensional outline of what that shape might look like to represent an enzyme. So this big purple blob represents an enzyme, when in actuality the three-dimensional shape is better represented by this ribbon diagram. So if we go back here, we can see that this enzyme has a particular shape on it. Generally, there will be a part of the enzyme that has a specific shape called the active site. And the reactant or substrate, which is your starting molecule of a reaction, is going to be bumping around just like all molecules are moving around in solution, and they'll go faster if the temperature is warmer. Sometimes one of those substrates can bump into the active site on the enzyme. And once they are bound together, the enzyme and substrate form what is called an enzyme-substrate complex. The enzyme will sort of squish the substrate around and align it in ways that make it easier for the substrate to make new bonds and turn into products. And then the products can leave the active site of the enzyme and go off and do whatever their function is in the cell. The enzyme sticks around, it's not broken down, and it's free to work again. So if another substrate were to bump into that enzyme, it could convert it into products and repeat the whole process over again. Here we can see that same process and how it fits with a written reaction. So if sucrose was our substrate, that would be like the yellow molecule. The enzyme sucrase that breaks it down would be our big purple molecule. Generally, the enzyme is going to be much bigger than the substrate. And the products that are formed are glucose and fructose, which are two smaller sugars, monosaccharides. So here our sucrose is moving around. One of the sucrose molecules bumps into the active site of the sucrase enzyme. The enzyme aligns and squishes it in ways that break the sucrose into glucose and fructose. The glucose and fructose can leave the enzyme, and now the enzyme could bind to more sucrose and do the whole process over again. So now that we know how enzymes work, let's talk about some things that can make them faster and increase the reaction rate. Enzymes are generally not in a solution where there is only one enzyme and one substrate. There are often many different molecules floating around in the solution, and they're all moving around. Faster or smaller molecules will move faster than larger molecules. So one thing that we can do to increase the reaction rate is to increase the enzyme concentration. You can imagine that if there's more than one of the same enzyme in solution, you will have more collisions between the enzyme and the substrate, which means more substrates turning into products. Also, if you were to increase the substrate concentration, again, that's going to increase the number of collisions between the enzyme and the substrate. Another thing is that if there are other molecules in the solution, like these green molecules, they could bump into the enzyme and sometimes block the enzyme and substrate from getting together. The enzymes and substrates could bounce off of those extra molecules, and it just reduces the chance that you have a collision between the two correct molecules. So if we remove those green molecules, anything that's not the enzyme and the substrate, that can increase our reaction rate too. Some other things that affect the enzyme reaction rate would be temperature and pH. Every single type of enzyme is going to have an optimum temperature and an optimum pH at which that enzyme works the best. And the reason it works well at that temperature and pH is because that's the optimal conditions for all of the charges on the various amino acids to line up so that the enzyme will be the right shape. In these graphs over here, we can see that the relative enzyme activity for the green enzyme increases from pH 1 up to about pH 2.5. So pH 2.5 would be the optimum pH for that particular enzyme. And if you were to increase the pH even more than that, the enzyme activity would get lower and lower as the enzyme unraveled or denatured and lost its normal shape. On this picture of an enzyme here, I tried to show what denaturing might look like, where the enzyme starts to unravel and that chain of amino acids comes apart and loses its shape. So now the active site is not going to be a very good fit with the substrate, and the enzyme is not going to be able to catalyze that reaction. The enzyme that works at low pH right here is going to have a different optimum than this other enzyme with the blue line that works better at high pH. This is actually the situation that we get in with stomach versus intestinal enzymes. The stomach has a very low acidic pH, and enzymes that work well in the stomach, if you were to move them into the intestine where the pH is more like 8, those enzymes from the stomach would denature and they wouldn't be able to function very well. Many bacterial enzymes work at pHs that are close to 7, which is neutral. And so when those bacteria, if you were to eat some of them in your food, when they get to the stomach acid would denature those bacterial enzymes and kill most of the bacteria that you come in contact with from your food. Each enzyme also has an optimum temperature. So in this lower graph, you can see that we have an enzyme that works best around 15 degrees Celsius, which is the orange line. And we have another enzyme that's more like what human enzymes would be like, where it works best around a temperature of 37 degrees Celsius, which is about 98 degrees Fahrenheit, normal human body temperature. Only bacteria that have enzymes with similar optimum as human body temperature are capable of infecting humans and causing disease. Bacteria that live in, say, hot springs, which are much hotter than human body temperature, are going to have enzymes with very high temperature optima. And those bacteria will normally have their enzymes denature and be unable to function if they were to get inside of a human body. Another thing to know about enzymes is they're extremely specific. The fit of an enzyme's active site with a substrate is similar to the fit of a particular key with a particular lock. Only particular kinds of substrates can fit with certain kinds of enzymes. Here are a few examples of different kinds of enzymes. Rubisco, this one that you can see spinning to show the three-dimensional structure, is an enzyme found in plants, and it was one of the earliest enzymes discovered, which is why it doesn't have the ASE at the end of its name. This enzyme takes CO2 out of the atmosphere and adds it onto molecules that later become sugars in photosynthesis. So it's an extremely important enzyme for photosynthesis, which keeps plants alive, brings them enough energy, which ultimately feeds animals and feeds the world. And also, if we could figure out ways to make Rubisco and other enzymes involved in photosynthesis more efficient at taking carbon dioxide out of the air, that might be useful in researching ways to combat climate change. ATP synthase in the middle here, this is an enzyme found in almost all living things, if not all living things. It spins like a turbine and adds two things together, ADP and phosphate, to make a molecule called ATP. And ATP is like the energy currency of the cell, so it's like money. Most of the activities that a cell needs to do, like building new molecules, like actively transporting things into places where they're at higher concentration, like moving muscles, each of those is going to have an ATP cost, where it costs a certain amount of ATP energy to move your muscles for a certain amount of time. And this ATP synthase enzyme is constantly churning out thousands and millions of ATP molecules all the time. Cellulase is another interesting one. This breaks down the polysaccharide cellulose, which is found in plant cell walls, and wood in trees is mostly made of cellulose. And that cellulase enzyme also has some commercial applications. People are starting to use it in making biofuels, breaking down plants. It's also used in making clothing to help soften fabrics and for instance, if you've ever had any clothing made out of bamboo, to break down the bamboo fibers so it's not so woody and gets to the point where it's soft, it involves that cellulase enzyme. And animals that eat a lot of grasses that are full of cellulose have bacteria living in their digestive systems that produce this cellulase enzyme. So things like cows and buffalo and other ruminants. So if we want to look at a few specific kinds of enzymes, here are some of the digestive enzymes that are active in the small intestine. All three of these enzymes, amylase, lipase, and trypsin, are made by the pancreas, which then secretes them through a tube into the small intestine. So the small intestine is another tube, and on the inside of the small intestine you have these folded structures called villi. Villi is the plural form of a singular villus. And the villi are covered with cells, so those little square structures are supposed to be cells. And the cells have these little tunnel-like transport proteins that can allow small food molecules to go inside. When you eat food molecules, they tend to be very large polymers made of many different monomers connected together. So these digestive enzymes are going to break up the large food molecules into smaller food molecules. If your food molecule was starch, a polysaccharide, you would use the enzyme amylase to break it up into monosaccharides and disaccharides. If the food that you had just eaten had a lot of lipids, which are things like fats, triglycerides, then the enzyme that breaks those up would be called lipase, and lipase is going to break those triglycerides up into fatty acids and monoglycerides. And then the enzyme trypsin, which is another one that doesn't end in ASE because it was one of the earliest one discovered, is going to break up long chains of proteins into individual amino acids and sometimes dipeptides, which is two amino acids connected together. Once the digestive enzymes break the food molecules into small enough pieces, those small pieces can then go through transport proteins to be absorbed into cells and then be transported into the bloodstream, which is like a highway that transports those nutrients all over the body. I hope that this was helpful and that you understand enzymes a little bit better now.